Hey, this is a video for Biodiversity.3. So go ahead and check out the essential vocabulary, set up your guided notes, and check out the aim. Describe how scientists can determine relationships between organisms. So just to review, we discussed cladograms before, and that is showing evolutionary relationships, where you can also see a common ancestor. And when organisms have similar characteristics, it indicates that they very likely had a common ancestor and therefore developed similar traits. So this term homologous structures is related to that. These are similar structures that are shared between different organisms because of a common ancestor. Some examples of homologous structures would be the forearms of a human, the front leg of a horse, front leg of a cat, the wings of a bat, the feathers of a bird, and the fins of a whale. If you notice, all of these body parts look different, but they all had similar origins, and therefore they're going to have similar number of bones and relatively similar structures. So why do related organisms have similar structures? Okay, one way that scientists can determine relationships is by looking at chromatography. And this is a laboratory procedure that separates pigments in a solution. So for example, if we took a solution and we took some paper in the liquid, we put in a dye or a pigment. That could be pigment from a plant, for example. The pigment is going to have different proteins. And what happens is the water soaks up as we go up this filter paper. Different parts of the water or different parts of the pigments will be dropped off. So as the water moves up, it brings with it pigments. Much like if your book bag gets wet during a rainstorm and you might have a pen on like a worksheet and that pen might smear all over the paper. That's kind of the same thing that's happening here. And then what you're going to do is you're going to test your unknown sample with other samples. And the ones that are the most similar or with the similar banding patterns are very likely the most related. So this process will work with plants. I wonder, can you answer, why do related organisms have similar color patterns? And now gel electrophoresis. This is a process, a laboratory procedure that uses electricity to separate DNA segments based on size. So what happens is you cut the DNA, you add electricity to it, and the DNA moves through this gel. It's basically like having a giant, not a giant, but like a little square of Jell-O. And when you cut the DNA segments, they're broken up into tiny pieces. And the smaller ones move the farthest through this gel. So the smallest segments are going to be at the bottom, and the largest segments are going to be at the top. Now remember, the electricity causes the movement, and the size of these DNA segments will cause them to stop. So we use the electricity, and then what we're doing is once everything is completed, you will compare for similar bands. The one that has the most similar bands, if you look here, this is the original one, and we're comparing it. The one with the most similarity will be the one that is the most closely related. So if you look here, that would be species B. Okay, so what is the factor that causes the DNA to move in the first place? After you answer that, I want you to tell me what is the factor that causes the DNA to stop. So what I would like you to do is first respond to this aim. Describe how scientists can determine relationships between organisms. And now I'd like you to show what you've learned by completing practice problem set 3. I hope that this video is informative. I thank you for your attention, and I will see you soon.